I'm very happy to welcome our first speaker, Emmanuelle Passegué. Are you there?
I am. Can you hear me?
Yes, we can hear you. Thank you so much. And you are dialing in from Columbia University?
Exactly, New York City, where it's end of the morning here and sunny as well.
That's great.
Can you see my screen?
We can see your screen, so you can start whenever you're ready. Thank you so much for joining.
Fantastic. So let's get into it by starting by thanking the organizer for the invitation and saying, oh, sorry, I am to not be able to be with you in this beautiful place in Copenhagen. So what I would like to tell you is really what we have learned on cell intrinsic and extrinsic mechanisms driving hematopoietic stem cell aging, and how we can take this information to try to rejuvenate all the hematopoietic stem cell function. So when we talk about HSC aging, it's really what's underlying a lot of these age-related blood defects that we are all familiar with. This decrease in red blood cell, anemia, and decreased production of lymphoid cell, increased production of myeloid cell, higher predisposition of blood cancer, and so forth. So all of that emerged from a dysfunction at the level of the blood-forming hematopoietic stem cell, which is shared between mice and humans. So looking in young mice, which are between 2 months of age, and comparing them to all mice, which are really old. Here we are talking about 24 to 30 months of age geriatric mice. We can see this massive expansion of the hematopoietic stem cell compartment investigated by flow cytometry and quantified here by frequency in the bone marrow cavity, where until midlife, there is not so much change in the number of all stem cell, but past midlife, toward 18 months of age, and even more so by 24 months of age, we have this massive expansion of the compartment, which become very dysfunctional in its regeneration potential that can be measured by transplantation, where we engraft in young recipient mice the same number of either young or old stem cell, and all stem cells are very low in overall chimeras, and their lineage bias is very obvious, with essentially production of myeloid cell, lack of production of B cell and T cells, the whole lymphoid compartment. So a lot of work has been done in the field trying to understand what drives this deregulation from the standpoint of cell intrinsic damage to the machinery controlling normal cell function, as well as what factor contribute from a dysfunctional microenvironment niche in the bone marrow cavity where the stem cell reside. So our lab has contributed to both aspects, focusing on various cell intrinsic damage like replication stress, autophagic regulation, and metabolic reprogram of the whole stem cell. But really what I want to tell you today is what we have learned and what we can do when we take this old hematopoietic stem cell from a 24-month-old mice, which have this damage to the DNA and other epigenetic change, and live in this remodeled inflamed bone marrow niche microenvironment. So the first approach we use is a very classical in the aging field, which is parabiosis, and really address the power of systemic factor, blood-borne systemic factor, in either rejuvenating all tissue function or in inducing aging feature in a young animal, which is surgically linked to an old partner. So we team up with Sol Vileda to do this experiment, and we know the parabiosis approach work in our end. We see improved neurogenesis, better number of D6 positive neurostem cell in the dentist gyrus. The problem is it doesn't work at all in the blood system. We see unchanged expanded number of all hematopoietic stem cells in the whole parabiome. They still have compromised function. Please focus on the orange color here. They still have compromised function, and they are still very myeloid biased in their output following transplantation. So there is a lot of caveat associated with the parabiosis. So very often these experiments are complemented with a plasma transfer where plasma from young mice are injected in old mice and can recapitulate the rejuvenative effect of having young blood into an old animal, at least has been shown to many other organs. And again, we know this approach works because we see rescue of neurogenesis in these plasma-injected old mice, but there is still no effect on the old hematopoietic stem cell with expanded number, decreased engraftment, myeloid bias, reconstitution, so no change. So this is functionality of the cell, and maybe we were looking to a too big step for this rejuvenation. So we then look at some intrinsic hallmarks that we have characterized in replication stress. And a characteristic feature is this accumulation of gamma H2AX at the nucleoli of all stem cell. And this mark is not changed in the whole parabiome stem cell. The expression of NCM is still decreased, and that's what drives, in large part, the replication stress feature in all hematopoietic stem cell, and their division kinetic are still impaired, which reflect this really strong impairment of their replicative activity. In addition, we saw no change in the degraded bone marrow microenvironment, here focusing on the endosteal population, where we can isolate endothelial cell, mesenchymal stromal cell, and various osteoprogenitor. These osteoprogenitor number are decreasing in all mice, still decrease in the whole parabiome, and their functionality in forming colony is as well largely impaired, and that's not rescue either. So no functional change in the whole stem cell, no change in their deregulated biology following exposure to young blood and young blood circulating factor. So with this result, we then turn to the crossover aspect of this parabiosis experiment, where you have cell migrating from one parabiome to another one. And it's very clear that there is equalization of blood crossover between the two parabiomes, with as much old blood cell being present in the young parabiome that you have young blood cell in the old parabiome. The crossover is less marked, less pronounced in the bone marrow, but it's still fairly balanced. What's very different is the crossover in the stem cell compartment, where there is way more old stem cell migrating in the young marrow than there is young stem cell migrating in the old marrow. There is no young cell. In the old mice, there is a large influx of old stem cell. And these old cells that you can barely see here with this blue color are not adding to the young stem cell in the young bone marrow. They are actually replacing young stem cell. And it's a very stem cell specific feature, because as soon as we look at the downstream progenitor, we are losing this chimerism, indicating that only old stem cell accumulate in the young bone marrow. So this leaves us with a perfect paradigm where we can study the naturally arming of old cell in a young marrow niche, and to see if the exposure to the young niche can have a rejuvenating effect on this old stem cell. And the result, again, were utterly disappointing. Yes, we have old cell in a young marrow, but functionally, they are still impaired, and their lineage output is myeloid biased. In addition, we still have the feature of replication stress, no change. And molecularly speaking, these old cells which lodge in the young bone marrow niche are still old. They have exactly the same difference of gene expression compared to young stem cell that we see in every other comparison, young versus old, young eyes or old eyes or young head, old head. This is exactly the same that we see here. They are as well different from the old cell, which remain in the old niche, in the sense that they are more metabolically active. They have more activation of stress response pathway and homing and migration mechanisms. So they are migratory old cell. They are not rejuvenated old stem cell. One feature of stem cell functionality is that when they get to a new environment, they do not acquire immediately restores their functional quiescence, which is important for transplantation. So maybe here we have an effect of early immigrant, and we need to wait longer for them to function better. So to assess that, we separate our parabiosis, and we kept these young mice, which had this old cell into their bone marrow niche for an additional 4 months to study whether it will have a better effect in turning back the clot and rejuvenating this old stem cell. And again, the result is no. Very quickly, the chimeras decline in the periphery, stay in the bone marrow, so they persist. They tend to expand, but their engraftment is still compromised, and their output is still myeloid biased. So no functional restoration of all stem cell function through this parabiosis, either by circulating blood factor or by long-term exposure to a young bone marrow niche. And in fact, we found a similar result with a whole range of systemic intervention. Exercise in a cohort which has been characterized for a very clear rejuvenative effect on neuronal stem cell, improved memory, has absolutely no effect on HSC function. Caloric restriction, lifelong with the NIA caloric restricted Black 6 cohort, same thing. Absolutely no functional recovery of defective old stem cell function, really leading to the conclusion that these old stem cells are past a point of no return. They are refractory to this systemic classical intervention, and that we need to do something else to try to improve their functionality. So this work has been published now in Journal of Experimental Medicine, and what I want to tell you is a much more positive spin on HSC rejuvenation, which still now to what we can do to target cell extrinsic mechanisms contributing to defective whole blood production. So I alluded to that, but now give you more detail about the remodeling of the old bone marrow niche, where compared to the young niche, which have a very dense set of population at the endosteum, mesenchymal stroma cell, osteoprogenitor, various endothelial cell, arterially central marrow sinusoidal, and a really small population of leptin receptor MSC, which wrapped around sinusoidal blood vessel. So that's a niche in the young. Completely changed in the old. Significant loss of all the endosteal population, degradation of the blood vessel network, massive expansion of this MSCL, which become very inflammatory. And as a consequence, we have low chronic level of inflammation in the bone marrow niche cavity with increase in the production of the very classical pro-inflammatory factor, interleukin-1, TNF-alpha, and so forth. So we know that this inflame and remodel old bone marrow niche actually drive activation of what we call emergency myelopoiesis pathway and reprogramming of the blood system in this old blood system with constitutive activation of a myeloid differentiation program, metabolic activation of all stem cell, overproduction of various multipotent progenitor, defective regenerative response. So all of that are intermingled dysfunction. All niche, all blood system, in the middle, you have the inflammatory mediator. So an interesting feature is that there is a lot of similarity between this age chronic inflamed bone marrow fluid and the senescence activated signature, which has been well characterized. However, we do not see any evidence of senescence in our age trauma, either by beta-activated SA-beta-gal or by single-cell RNA-seq with no activation of senescence pathway. What we know is that when we inject chronically interleukin-1 by daily injection in a young mice, we model aging feature. We model the reprogramming of the blood system with increased myeloid cell production at the expense of lymphoid cell production and red blood cell production driving anemia. And we as well remodel the niche in these young mice with similarly to the effect of physiological aging, expansion of dysfunctional vasculature with cell lost by flow cytometry and degraded endosteal population. So We know as well that IL-1 is essentially produced by this degraded bone marrow niche. We have our result of our SMART-seq sequencing, which show, especially in this osteoprogenitor-like population, much increased expression of all the IL-1 signaling pathway. We confirm that by qPCR, and by caspase-1 activity, we read out really processing of interleukin-1 from this damage on osteoprogenitor population at the endoscope. So the niche produce IL-1. And basically, by targeting IL-1, we can really improve both the niche and the whole blood system. So how we did that? We use anakinra, which is an FDA-approved antagonist of human interleukin-1. It blocks IL-1 signaling. We cannot give anakinra too long to a mice because you induce an immune response, but we can do short-term treatment, which allows us to look at the blood system. So I don't want to take you through all these data. They are complex, they are specialized. It's really hematopoiesis 101. But what I want to say is that we give to anakinra for 2 weeks. We do not change steady state hematopoiesis. We give anakinra for 2 weeks with a stress challenge and using hematopoietic regeneration. And we can very significantly improve regeneration in all mice. Focus mostly on the picture of the bone marrow cavity. All mice damaged with 5-FU chemotherapeutic compound, it's very nasty. There is also mostly megakaryocyte and very bad-looking central marrow. You give anakinra, you block interleukin-1 signaling, you restore blood production, you recover some of the anemic feature, more red blood cell, more lymphoid cell being done. So a better regenerative response. You block lifelong interleukin-1 signaling in interleukin-1 receptor deficient mice. You really significantly delay stroma aging feature. Again, I'm not taking you through all the data, but basically we dampen considerably this inflammatory MSCR, leptin receptor mesenchymal stromal cell compartment, that we can see by looking at the population by flow cytometry or by doing various types of single cell RNA-seq, with really a reversion of all the deregulated inflammatory pathway that we normally found in all mesenchymal stromal cell. And I see the moderator coming, so I assume I am out of time. So very quickly, last result side, the blood results are improved as well, with recovery of some of the hematopoietic population, change in the activation level of the stem cell, and better engraftment of this IL-1 receptor deficient whole stem cell. So putting that together, we know from this work that we have no functional rejuvenation by classical systemic intervention. We need to be smart. We need to know what's happened really in the bone marrow affecting this whole hematopoietic stem cell. And we can have a very nice functional improvement by targeting niche-mediated inflammation, which restore some parameter of blood production, not everything. So we need more, but we certainly need that to really help the elderly to fight infection and maintain proper immune response. And with that, I want to stop by thanking my lab, which did all this work, previous generation of training, which have left, a new generation of training, which finally had their first lab outing, nicely socially distanced on Fire Island this summer, lots of collaborator funding and resource. Thank you for your attention. I hope I have time to answer a few questions. I don't hear you. I'm not hearing anything. Sorry.
Very embarrassing. This was my mistake. I was muted, not even on Zoom, but on my microphone. So we have a question here from the audience. Rudy Westendorp.
Yep. So the bone marrow failure is, if I understand you well, is because of an expansion of dysfunctional old stem cells.
So I clarify what you mean by bone marrow failure. So the whole stem cell compartment is expanded. So there is more stem cell, but on the cell by cell basis, they are much less good at producing blood.
Yeah.
So
my question to that one is, let's say, is it a clonal expansion or is it, let's say, an expansion that is, let's say, more heterogeneous and perhaps driven by inflammation? Because that is what, let's say, I sense between the lines.
Yeah, so there is clonal expansion. So with age, the clonality of the pool contracts, so much less stem cell productively contribute to blood production. Why? Because of mutation, but as well the role of inflammation, which really change the output of this whole cell and focus it to production of myeloid cell at the expense of immune cell and red blood cell. So it's a compound mixture of different features resulting in impaired whole blood production.
Very nice. We have one last question. Please keep it short. Thank you.
Thanks for a very provocative talk. I'm just wondering, with so many elderly people on this treatment for osteoarthritis, has anyone looked to see what's happening to their stem cell niche?
No. And I mean, not as I know. And I hope that when we finally publish our work, which we are resubmitting soon, it will drive a lot of investigation of this cohort. I think it will be fascinating. Investigating the niche in human is complicated, but I think it's very exciting. Yeah.
Thank you.
All right, thank you very much. And let's give Emmanuelle Passegué another round of applause. Thank you so much for joining.